Start our conversation with a discussion of general points of consideration when you're selecting antibiotics. And then we will dive into some mechanisms for various antibacterial agents. So what are the general considerations? So first, the source of antibiotic. It can be natural. Remember, antibiotics are chemical weapons that various bacteria use to kill each other. It's normal during the competition. Antibiotics can be synthetic, therefore produced by humans, derived by humans. And antibiotics can be semi-synthetic. Those are the ones that used to be natural, but then humans modified it. Okay? Spectrum of action. You want to use broad spectrum when you treat something that you don't know what it is. So you cover all the bases. Or if you know the exact positive agent, you may consider narrow spectrum. If you treat an infection in a patient who is otherwise healthy, then maybe a bacteriostatic antibiotic would work. But if you have to deal with a patient who is immunocompromised and whose immune system is kind of down, you may need to consider bactericidal. Now, if treatment can be done in the outpatient settings, or is done in outpatient settings, antibiotics preferably should be oral. But with the patients who are in the clinic, in the hospital, intramuscular or intravenous delivery can be possible as well. And you may wonder, yeah, that's kind of duh, but some antimicrobial agents are not readily absorbed in the gut, so they cannot be used in the outpatient settings. You need to consider whether the resistance to a selected antibiotic is common. And not only this, you need to consider if your selected antibiotic can cause what we call a superinfection, when it wipes out a good microbiome, okay, leaving behind some resident pathogenic bacteria that can cause things like Clostridium difficile infection or bacterial or fungal vaginosis. So far, make sense? We're good? Okay. We're probably going to be adding some more points here, but we're going to leave them here for now, and we will dive into the mechanism. Now, I'm going to classify antibiotics based on their cellular targets. Okay, so antibiotics can sort of target different cellular processes. And the first target that we're going to be discussing is cell wall. More specifically, cell wall synthesis. So there are three broad categories of cell wall synthesis inhibitors. The first category of cell wall synthesis inhibitors are beta-lactam antibiotics, beta-lactams. So what is common between all those antibiotics? They all inhibit basically same step in the process of the cell wall synthesis. So I want to remind you that cell wall consists of peptidoglycan. I hope that term means to that. Okay, peptidoglycan. Peptidoglycan is the cross-linked polymer that is produced when glycan chains are cross-linked, connected by peptide linkages formed by amino acids. And this is not a reaction, this is more of a process, right? Okay, so this is the process when glycan chains are linked together by peptide linkages derived from amino acids, you have peptidoglycan cell wall. Okay, good? The enzyme that facilitates this process is called transpeptidase or penicillin binding protein. So beta-lactams essentially inhibit transpeptidase. They stop it from working. And they do so because they have a common structural feature. Beta-lactam ring. Does that make sense to you? Does that make sense? Good? Okay. It's kind of reiteration. All beta-lactam antibiotics have beta-lactam ring. This beta-lactam ring enables them to inhibit transpeptidase, thus stopping formation of cross-links. So glycan chains remain non-connected. Does that make sense? Not connected. And since they are not connected, cell wall is mechanically weak and cell just blows up. So far, we good? So let's see what are main groups of beta-lactams. These main classes are penicillins, cephalosporins, monobactams, and carbapenems. All these four classes have beta-lactam ring. Does that make sense? They have different side chains, okay? They are different from each other, and that difference, those different side chains that are added to beta-lactam ring changes the spectrum of action, the bioavailability, allows them to overcome the resistance. For instance, moxicillin is the oral penicillin drug. Ampicillin is the IV or IM penicillin drug. It is not readily bioavailable in, via the oral. So far, are you following? When we will wrap up cell wall synthesis, I'll explain what you need to know. Deal? Okay. So a couple more classes here. Another class is glycopeptides. And most famous representative here is vancomycin. So glycopeptides inhibit both peptide link formation. And ligand chains formation. Okay. And the third category here, just one antibiotic, beta-Lactamase, which inhibits transport of cell wall subunits across the. So, first of all, let me kind of give you an analogy which I think is useful. If you think about cell wall as being built from bricks, well, bricks and mortar, then all beta-lactams get rid of mortar. Make sense? Mycobacterium get rid of mortar and also put bricks into disarray. And beta-tracin stops the trucks that carry brick and mortar to get to the building site. Does that make sense? We're clear. What do you need to know? 1, 2, 3, 4 beta-lactam antibiotics, glycopeptides and beta-tracin, 6 classes here. Now, do you need to know individual antibiotics? For instance, methicillin or ceftriaxone? No. I will not ask you the names of individual drugs. I'm interested in classes here. Does that make sense? Okay. Now, I want to outline one very interesting and quite frankly, very important mechanism of antibiotic resistance. Okay. And actually, I just thought about a couple more considerations here, which we will add. I already forgot one. So let me outline again mechanism of action for beta-lactams. I'm going to just write it down. So you've got glycans, right? Plus amino acids, and you have cell wall. Now, what facilitates this process? B. Correct? Now, beta-lactams, what do they do to PDP? They inhibit it. Does that make sense? And I specifically, sorry, put this sign with a horizontal line at the end of it. That's a typical symbol that is used to show that it's inhibition. Okay, make sense? But here's an interesting story. Some microorganisms produce an enzyme that is called beta-lactamase. And beta-lactamase doesn't inhibit beta-lactams, it destroys them. Okay? So beta-lactamase destroys the beta-lactam ring, and if beta-lactam ring is gone, beta-lactams do not work. Are we clear? Now, what can we do about this? Turns out there is an approach, a drug called flavulonic acid inhibits the enzyme beta-lactamase. So beta-lactams inhibit PBP. Beta-lactamase destroys beta-lactams, rendering them useless. However, if you use clavulanic acid, it will inhibit beta-lactamase. So beta-lactams are functional again, and therefore you restore the sensitivity of microorganisms to beta-lactams. Does that make sense? Usually beta-lactamases are called like penicillinase, cephalosporinase, carbapenemase, but you get the gist, right? So far, good? Now, yeah, the drug, by the way, you may have heard about it. It's called Augmentin. It's amoxicillin plus clavulanic acid. Now, I want to ask you a pretty simple biology question. Well, seriously simple. So bacteria have cell walls, right? Well, not all of them, but many of them. Human cells. Do they have cell walls? Huh? What's that? Good job. No, they don't. They don't. Absolutely. They don't have cell walls. Therefore, these drugs have nothing to inhibit in human cells. Does that make sense? So this concept is called specific toxicity. We want antibiotics with the high specific toxicity. We want antibiotics that affect only bacteria, but not humans. Does that make sense? We're good? Moving on? Okay. Next set of targets here is protein synthesis. Okay. Namely ribosomes, and you will see. So in terms of the protein synthesis targets, we can identify three main types of inhibitors. First type is inhibitors of the 30S ribosomal subunit. Second target is 50S ribosomal subunit. And the third target is initiation complex, and I'll elaborate on that. So, what are the 30S subunit inhibitors? There are two groups of drugs in this category. Look, one is aminoglycosides. Aminoglycosides, they distort the structure of 30S subunit, which leads to the production of faulty proteins. And accumulation of faulty proteins eventually kills the cell. So far, we're good? Second class here is tetracyclines. This is almost a household name among antibiotics. Tetracyclines inhibit amino acid transport of tRNA, basically preventing tRNA from binding into the A site. So far, are we good? Now, what about 50S subunit inhibitors? There are two large groups. One group contains streptogramins, macrolides, and glucosamides, which all inhibit the release of a polypeptide from the ribosome. Essentially, nothing dies, just polypeptide can't leave ribosomes, and all ribosomes are just completely occupied and busy, and so protein synthesis stops. Now, second category here, the group of antibiotics, well, actually, it's like one kind of generic name, chloramphenicol. So chloramphenicol inhibits addition of new amino acids. Basically, it inhibits the enzyme called peptidyltransferase. And finally, in terms of the initiation complex inhibitor, a group of drugs called oxazolidinone. Okay, now, let me elaborate on some of this stuff, right? So think of a ribosome. You have small subunit, which is 30S, large subunit, which is 50S. If you target 30S subunit, aminoglycosides will be distorted, and that will lead to bad proteins being produced. Tetracyclines prevent amino acids carried by tRNA to be transported to the ribosome. Okay, we good so far? Makes sense? Now, sort of names, aminoglycosides, for instance, kanamycin, tobramycin, tetracycline, doxycycline, 50S subunit inhibitors, streptogramins, macrolides like azithromycin or ZPAP, glucosamides like clindamycin. Okay, they inhibit the release of polypeptide. And chloramphenicol is a synthetic antibiotic that inhibits addition of new amino acids. Now, in this case, oxazolidinone. So what I want to kind of highlight, these groups, all of them, inhibit the process of translation. Translation has already started. Oxazolidinones do not let translation to even start. They prevent ribosome from assembling. Are we clear? Does that make sense? Now, again, those are the classes that I want you to know. Now, we already kind of highlighted that cell wall synthesis inhibitors have high specific toxicity, right? Now, remember that bacterial ribosomes are 70S ribosomes, and human ribosomes are 30S. Based on this information, what do you think about the specific toxicity of inhibitors of protein synthesis? Is it going to be high or low? If human ribosome is 80S and bacterial is 70S. In other words, human ribosome is different from bacterial. What do you think about specific toxicity of those? It's 50-50 question. Nothing to lose. Huh? It's going to be high because it's high specific toxicity. It will preferentially inhibit bacterial ribosomes without targeting so much human. Does that make sense? Because ribosomes are different. So also, bispecific toxicity. Okay. I'm going to take a short pause and ask, so far we're good? When I will fill all allocated space, we'll take a pause so you can take a picture, if you want. Okay. So let's get to another target here, which is going to be cell membrane. In this situation, in the cell membrane situation, we have two groups of antibiotics, which are, in my opinion, pretty cool. One group here is called polymyxins. And another called lipopeptides. So lipopeptides form pores in the membrane. While polymyxins, they destroy the membrane structure. I have another question. What is membrane, a membrane, cellular membrane, made for? What is it made of? What are the molecules that make up the membrane? Anyone? The pH word. Huh? No. Phospholipids, right? Bacterial membrane is made of phospholipids, and human cell membrane is made of phospholipids. What do you think about specific toxicity of those antibiotics? Yep. Let me demonstrate how low it is. Lipopeptides are not used very widely. Polymyxins. Polymyxin B is so toxic that it is used exclusively on the skin and wounds. It is not used as a systemic drug. IV, IM, oral, no one. Does that make sense? Its brother or sister, whatever, called colistin, is used for procedures like wiping out all your gut microbes. So you can imagine, or treating multi-drug resistant systemic gram-negative infections. So basically, colistin is a choice of desperation. Does that make sense? Side effects are so massive that those drugs are used topically and very infrequently systemically. Does that make sense? You can basically say your kidney is goodbye if these drugs are used. Okay. Now, next group of drugs. I won't have a space for just one group. I maybe will deal with that. Next group of drugs, the inhibitors of nucleic acids of various sorts. So, what is here? We've got inhibitors of DNA synthesis. Okay, this is going to be fluoroquinolones, and that's basically replication inhibitors, right? And that's fluoroquinolones. Okay. Now, fluoroquinolones inhibit the enzyme called DNA gyrase. That should sound familiar. They are super effective, but have kind of, a bit of a side effect because DNA replication is quite conserved between bacteria and humans. Then we have inhibitors of RNA synthesis. Or transcription. So this is going to be rifamycins. And they inhibit RNA polymerase. And finally, inhibitors of DNA repair. Now, I'm going to put 3 question marks here. Because for these drugs, we don't really know what the exact mechanism is. It looks like it's the DNA repair inhibition. Those drugs are called nitroimidazoles. Okay, now, let me give you some examples. Have you heard of ciprofloxacin? Cypro. Anyone? Heard? No? Oh, lucky you. Cypro is a great broad spectrum, very efficient antibiotic that can completely ruin your connective tissue as a side effect. Okay, that's fluoroquinolone. I hope nobody have heard about rifampin. No? Good for you. It's a drug to treat tuberculosis. And have you heard about metronidazole or flagyl? Anyone have a clue? Go? Okay, well, those are lucky. Nitroimidazoles, namely metronidazole or flagyl, they are interesting drugs. They've been around for a long time, and they inhibit basically all anaerobic microorganisms, not only bacteria, but also protozoa, for instance. So why I, say a dog, when a dog has colitis, like diarrhea and stuff like that, like bloody diarrhea, flagyl will fix it. Basically, just wipes all the bad anaerobes out of the skin. Okay, make sense so far? Let me pause the recording so you can take a picture if you want. So next target that we're going to talk about is metabolic processes. There are, yes? It varies. I mean, Cypro, usually used short term. The toxicity accumulates with time. For colistin is just colistin is awful, just plainly awful. It's like this patient, if we don't give them colistin, they're gonna die anyway. So let's just give colistin, maybe they will survive, and then we'll deal with the fallout later. Does that make sense? Cypro, for instance, it is often used for ear infections and eye infections because it's not systemic. And actually, thanks for bringing that up. So you don't want to use that nuclear weapon first. Does that make sense? Nobody starts treatment of sore throat with carbapenems. It's going to be penicillins first, okay? Nobody starts using, well, colistin for gram-negative infection of urinary tract. It's going to be cephalosporins first. Does that make sense? I had a student with, well, kind of TMI, but urinary tract infection. And they started with cephalosporins, then they moved to, it was apparently gram-positive, then they moved to, I think, azithromycin, then they moved to something else because it's just, it's resistant, okay? But you get the gist, right? So, metabolic processes. That's going to be different in terms of the specific toxicity, kind of a word of word. So first group of inhibitors, I'm actually going to do it slightly different order. So the first group of inhibitors that I'm going to put here in the corner, isoniazid. So isoniazid inhibits mycolic acid synthesis. Inhibition. Now, if you remember, we mentioned that a group of microorganisms called Mycobacterium, remember those? Mycobacteria. They have mycolic acid layer outside of the cell. We good? So isoniazid is specifically used to treat those, mycobacteria. And we'll get back to this. Okay, now, second group here, let me try to put down, it's diryl-quinolones. These ones inhibit ATP synthase function. Okay? So to Page's question, these ones, diryl-quinolones, are fantastic treatments against Mycobacterium tuberculosis, for instance. However, ATP synthase is quite conserved among all species. So if it's conserved among humans and bacteria, what can you say about their specific toxicity? If they are very similar. Love it, yes. They have terrible side effects. So low specific toxicity. And finally, this group here, there are two drugs in that group. It's sulfa drugs and trimethoprim. They inhibit folic acid synthesis, which results in the inhibition of nucleotide synthesis. So far, does that make sense? So folic acid is essential molecule to synthesize nucleotides. If folic acid is not synthesized, nucleotides cannot be synthesized. Am I clear? Does that make sense? Now, I'm going to outline something and then add another point of consideration. So, the pathway of folic acid synthesis starts with a chemical called para-aminobenzoic acid. Para-aminobenzoic, and I don't remember, there's some few steps. I quite frankly, I don't remember what those, what happens during each step. I don't remember the names of the enzymes. I think it's dihydrofolic and then tetrahydrofolic acid, but I wouldn't take my words to the bank, okay? So I'm going to put A and B as intermediates. So we've got intermediate A, then we've got intermediate B, and then here, other stuff here. So sulfa drugs, look, sulfa drugs, they inhibit the first step of the pathway, the first enzyme. And trimethoprim inhibits the second step of the pathway. Those are competitive inhibitors, okay? Bless you. So they competitively inhibit two different enzymes of the same metabolic pathway. And you may ask an absolutely legitimate question. Why the hell do we need two inhibitors of the same pathway? The answer is this. If you use either of the drugs by itself, these drugs are bacteriostatic. If you use them together, these drugs are bactericidal. This is called synergism. Does that make sense to you? Now, ever heard about the drug called Bactrim? Anyone, Bactrim? Does it sound familiar? It's so great to see people. And I'm kind of a little bit ironic that don't get sick too much. That's the advantage of being young. Okay. Bactrim is a good drug, pretty more against gram negatives, but kind of general broad spectrum antibiotic that works not only against bacteria but also against some protozoa, and that is sulfamethoxazole and trimethoprim combined. We're good? Clear? Now that's the only thing that was not in the original picture, so if you want to take a shot, go for it. So a few more things to discuss: resistance, testing of antibiotics, and Mycobacterium tuberculosis. So let's talk about antibiotic testing first. And when I say testing, I basically how you determine if a particular antibiotic is good to treat a particular infection. I separate methods for testing into two loosely defined groups. One group, well, it's not even a group, it's a method, called Pearson flower disc diffusion test. Now that's the test that you have done last week, remember? When I was dispensing antibiotics on your agar plates. It was, look, admit it, fast, easy. The only problem is each disc has antibiotics in a certain concentration, which is clinically relevant. The problem is you can't test any other concentration. You're kind of stuck with what you have. If that makes sense. So if you want to figure out what is the minimal concentration in which your antibiotic will inhibit the growth of microorganisms, you should use dilution tests. Either in the tubes, in the plate, or you can use E-strips, which is basically a strip of paper with different, progressively more and more diluted antibiotic, which kind of shows you the inhibition. So these methods, tube dilution test, plate dilution test, or E-strip, they all allow you to determine what we call MIC, minimal inhibitory concentration, the lowest concentration of antibiotic that inhibits the growth of a particular microorganism. You do serial dilutions, and then the lowest dilution that stops a bug from growing is your MIC. Does that make sense? Now, not only you need to know what is the lowest inhibitory concentration, you also should know at what concentration your antibiotic starts killing patients. Does that make sense? And this drives us into a concept of therapeutic index. It will be closely related to an idea of specific toxicity. This is why I put it so close. Now, what is therapeutic index? For each drug, you can define more or less two very important parameters. First is median effective dose. So median effective dose is a dose of a drug necessary to successfully treat 50% of experimental group. Does that make sense? It's not easy to determine, but it can be done. Okay? And then for each drug, you have median toxic dose. Okay? That's, well, quite logically a dose of a drug that causes toxic effects in 50% of experimental group. These two doses, these two parameters, do they make sense to you? Now, therapeutic index is a ratio between the median toxic dose and median effective dose. Does it make sense? Now let's do some math. How about that? I'm giving you numbers. So you have a drug for which median toxic dose is 200 milligrams per kg, and median effective dose is 4 milligrams per kg. What's the therapeutic index? Huh? 50, yes. You take 200, which is a median toxic, divided by 4, which is a median effective, you get therapeutic index. Now, 3 out of 10 questions. Which therapeutic index is better? Basically, which drug is better? With a higher therapeutic index or lower? Higher. Why? So I'm going to translate because it's all for recording. So what Paige said is higher toxic dose means that people are not getting affected as fast or as much. Absolutely, yes. You also have a wiggle room. If your toxic dose is 200 and effective dose is 4, you can give patient 12, the patient's going to be just fine. And you're right, if it accumulates, if it's not excreted, then you can sort of, give it more frequently, all that kind of stuff. Does that make sense? And yeah, there are all kinds of drugs, some drugs like, I don't know what therapeutic index for colistin, but I bet it's pretty low, okay? So drugs with a high therapeutic index are considered to be bad. Is that clear? Okay. Antibiotic resistance. I'm going to briefly outline some concepts here. So two categories, and microbiology has a lot of definitions that scientists are still quabbling about. So for instance, here, it's sort of my own definition. Resistance can be intrinsic. And it's not a great term, but I cannot come up with anything better. Acquired. So they all acquired, essentially. They all acquired in the process of evolution, right? But what I mean by intrinsic, if you go out there in the woods, and you collect a soil sample, and you isolate microbes, is there a chance that some of those microbes will be intrinsically resistant to, say, penicillin? Although they never infected humans or anyone. Yeah, because penicillin is a natural compound. It's a chemical weapon. So there's a chance that microbes in the environment have been exposed to penicillin and over the course of evolution develop this intrinsic resistance. Does that make sense? So basically you can say, yeah, we have like bacilli, like all bacilli, for instance. I don't know, I have no idea what I'm talking about, but bacilli, say, have intrinsic resistance to aminoglycosides, just all of them. Virulent, non-virulent, doesn't matter. That makes sense? Acquired is the resistance that was driven by humans. We use antibiotics in agriculture, we use antibiotics in medicine, and microbes evolve and exchange genes, and we drive this resistance. Does that make sense? Great example, methicillin-resistant Staph aureus with Staphylococcus aureus. Is it a pathogenic bacteria? Yes. Can you treat it with methicillin? Up to a certain point, you could, but then it became resistant. We clear? Okay. Now, mechanisms of resistance. I'm going to classify them by targets. No, not targets. No, better one. Just mechanisms. There are 8 mechanisms that I want you to know. Now, what do I mean by I want you to know? You should be able to match a description of the mechanism to a term. We good? So first category of mechanisms are related to drug. It's going to be drug modification. Okay. Inhibition of cellular uptake. And efflux pumps. Let me explain. You have beta-lactam with the ring. Are you following me? Beta-lactam with the ring. You destroy the ring, you modify the drug. Got it? You destroy the drug or change it chemically so it doesn't work anymore. Does that make sense? Inhibition of cellular uptake. You prevent drug from getting into the cell. Simple as that. You're not expressing some channels, something, reduce the number of channels. Efflux pumps. You pump the drug out of the cell. Any questions on that? Okay, yeah, this is pretty simple. Now, another mechanism is associated with the target. Okay, now let me clarify what I mean by target. Beta-lactams inhibit transpeptidase. So transpeptidase is a target. Rifamycins inhibit RNA polymerase. RNA polymerase is a target. Am I clear? So what can you do with a target? You can modify it so target doesn't work anymore. Okay, so no, pardon. The drug doesn't bind to a target anymore. Good? You can overproduce it. Try to get the dose high enough to inhibit a gazillion of targets. Okay? You can mimic target. Produce a molecular decoy. I find this method particularly exciting. I think I'll elaborate on that. So imagine you have a drug. Imagine you have a chloramphenicol, Cypro. You have a Cyprofloxacin, that binds to DNA gyrase and inhibits it. So bacteria will produce a molecule that chloramphenicol binds to instead, and it spares DNA gyrase from being inhibited. Does that make sense? And I'm going to put this here. It kind of doesn't really belong. It's enzymatic bypass. So what I mean here is you have to go from chemical A to chemical Z, right? And you do it in a certain enzymatic steps. So if drug inhibits one of those steps, then maybe with time, bacteria will develop the bypass around that step, that they can engage if the drug is around. Does that make sense? So, A, mini cells. This is acutest mechanism. It works against aminoglycosides. The study was actually published last year. So aminoglycosides cause accumulation of faulty proteins. So cells, what they do, they package those proteins into little blobs of membrane called mini cells, together with some antibiotic, and just get rid of it. Does that make sense? Like a tiny little trash can that they just, or trash bag that they just remove from time to time. We good? There is something I want to highlight, which is really important from evolutionary standpoint. All those mechanisms, they require some quote-unquote investment. Energy to pump out. Regulation of chlorines, maybe it will damage some other processes. Production of a special enzyme that modifies the drug. Production of a molecular decoy. Overproducing a target. So you see, it all comes at a cost. So it's not really paradoxical, but if you think about it, in the environment without antibiotic, microbes that are not resistant, listen, not resistant, they have fitness advantage because they don't have to waste the resources on these mechanisms. Does that make sense to you? Okay. Now, last bit before we take a break will be about Mycobacterium tuberculosis. And it's a pretty interesting story. I'm going to outline kind of, main features. That's a long name. So first of all, it's gram-positive, or ESA. I'm going to put gram-positive in the parentheses because based on the cell wall structure, it is gram-positive. But remember about the layer of mycolic acid outside. Remember that? Typical, thick peptidoglycan wall outside of the cell membrane, and then mycolic acid, which makes it, by the way, pretty resilient. It's slow growing arrow. Okay, really slow growing, and that will become really important. So let's tackle first transmission route. Respiratory transmission. Highly effective. Quite frankly, coronavirus is a child's play. Estimates put that 1/3 to a half of the world population are infected with Mycobacterium tuberculosis. However, infection often latent. Even if you are infected, it does not mean that you're going to develop a disease. Does that make sense? Only about 5% develop the disease. We clear? Transmission is super efficient, like extraordinarily efficient. But only about 5% exposed will develop the actual disease. Now, pathogenesis of the disease is due to the immune-mediated cellular toxicity. I'm going to break down this language. So what happens? Let me add one more detail, and then I'll explain it. It infects macrophages. So let's start with macrophages are the immune cells, right? Macrophages, big eaters that are supposed to destroy bacteria. Are we clear? So mycobacteria can infect macrophages and survive inside and actually live within the cell that is supposed to kill it. Immune system recognizes it and tries to destroy an infected macrophage. Usually, immune system fails to do so. So you have sort of an ongoing battle between immune system, mostly cytotoxic T cells, and macrophages, and the bacteria, okay? And eventually, you have the development of sort of a necrotic focus, necrotic core, with all the bacteria and macrophages in it, and T cells and whatnot. Eventually, immune system seals that necrotic core off. It forms a fibrin capsule that basically traps all that bad stuff inside, like in sarcophagus. Does that make sense? This is called a tubercle. So you may have a tubercle in the lungs or in the lymph node, and it will contain bacteria inside. But as long as your immune system is fine, you may not even show any symptoms. Does that make sense? When your immune system becomes a little bit depressed or something happens, bacteria spills out, since it's usually in the lungs, it all starts in the lungs, okay? It causes what's called chronic tuberculosis. And I want to highlight that all that crap that's going on with all that necrosis and dying cells, it's all because your immune system attacks, okay? So, pulmonary tuberculosis. Okay, Pulmonary chronic tuberculosis symptoms. I'm interested if what I'm going to say will sound familiar. Frequent cough, shortness of breath, night sweats, fatigue, low-grade fever. That's a questionnaire. When you get to a new physician's office, you can answer those questions. Basically, fatigue. Okay. Now, obviously, this is confined to lungs. You have a green sputum. You have maybe bloody sputum. Okay, that's all. That's all. By the way, you can stain for tuberculosis. If you stain sputum, it's Zil-Nielsen stain. If you stain in a culture, that's acid-fast stain. We'll get to the culturing. This microorganism is terrible because it's slow growing. It takes weeks, if not months, to grow it in the blood. Anyway, if it spills out of the lungs, it causes extrapulmonary or miliary tuberculosis. Prognosis is usually poor. The disease can affect bones, heart, meninges, kidneys, spinal cord, you name it. My grandmother's brother, grand uncle, I mean, it was a long time ago, 1938 or something. He died of the tuberculosis of the bone. It just happened there. Okay? Now, treatment is long and hard, up to 12 months. This is why there are so many drug-resistant strains, and there is a totally drug-resistant strains. There are totally drug-resistant strains of Mycobacterium tuberculosis. Because imagine, like, do you have, well, personal question. Let's say, okay, I have health insurance. God forbid I get TB. I have prescription insurance. My doctor prescribes me 12 months of antibiotics, and my insurance will cover. Okay, so yeah, I'm going to pay something out of pocket, but that's okay. Someone in Africa or India, they don't have financial resources to pay for the drugs for 12 months. So they will ask someone who has prescription privileges for, can you give me something shorter? Maybe 2 weeks. They say, yeah, sure, here's the slip. And they will go and get antibiotics and they will start taking it and they will feel better. But they will drive this resistance. I can't blame those people. It's a structural deficiency. You know what I'm saying? But 12 months is a long time. And that's an example. It's much harder to treat slow disease than fast disease. Because with a fast disease, you either cure it or the patient's dead. But you need to maintain a high concentration of drug over a short period of time. Here, 12 freaking months. Does that make sense? Are we clear? What about prevention? Well, hygienic methods like avoid overcrowding, don't put people in freaking jails. It's a jail disease. Okay. What about vaccine? It's called BCG. Very ineffective. Okay. So I know for sure that I am vaccinated against tuberculosis because in the United States, people do not get BCG because of low efficiency and because tuberculosis isn't, rampant here. Back in Russia, the goal of this vaccine is not to prevent infection. It is to reduce the rate of transmission and to prevent extrapulmonary tuberculosis in children. Does that make sense? So it's like, yeah, children's going to get it. Just let's make sure that they are not developing bad disease. They're just kind of mild. Okay, this vaccine probably is the worst one that was ever developed by humans. Okay, among the ones that are still used. Now, kind of a segue into any questions on TB? Oh yeah, it's a major cause of death among the infectious diseases in the world. 1.7 million a year. So every year. So when you look at coronavirus numbers, just remember, every year about 1.7 million people in the world die of tuberculosis. Okay. Last little bit, there's a page in the lecture notes with a whole bunch of acronyms. Acronyms like MRSA, VRE, CRE, ESBL, G-. Those are main terms related to common antibiotic resistant pathogens. Does that make sense? I'm going to be very clear in what I want you to know about this. You don't need to know the mechanism of resistance. Right? Is it a target modification or reflex sparks? This and that. You do not need to know, is it hospital acquired, community acquired, blah, blah, blah. You must know what the acronym stands for. So basically ask, in the acronym CRE, which microorganisms are we talking about? Can you tell me? In CRE, we are talking about Enterobacteriaceae. And what are those Enterobacteriaceae resistant to? Carbapenem. Carbapenem resistant Enterobacteriaceae. Or MRSA. What is the bug? Staph aureus. What is it resistant to? Methicillin. Only those that are in the table. Did I make myself clear? Any questions? Let's take a look.